Okay, hello everyone. Welcome to today's Journal Club. Briefly, we'll be discussing the biochemical role of the endocannabinoid system in the pathophysiology of attention deficit hyperactivity disorder. It's a narrative review and speaks and talks about some future directions. Next slide. Oh, this is introductions. Yep, I think PRC sponsors Journal Club. We have three pillars. Salus is our clinical research pillar. Buds Info is really our analytical pillar, and Cerium is our education pillar. Next slide. If you are new to our Journal Club, we do ask that you scan this QR code and fill out our questionnaire. We do use some of the information there to understand our our participants who join in Journal Club and helps us determine future topics. Next slide. All right, so leading the discussion today for Cerium Journal Club is Dr. Phil Malloy. He's a rheumatologist who is currently retired but continues to be very active in caring for patients. Dr. Jehan Marcoux, who's a cannabinoid researcher and owner of Delaware Analytical, which is a research lab. Dr. Kamen may or may not join us today. I know that he's just back and catching up and has a lot of patients today to see. He is the clinical director at Moss Rehab Outpatient Center at Jefferson University and also the president of the Pain Society in Pennsylvania and the head of the Rothman Opioid Foundation. Okay, I'm going to think the next slide. I hand it over to you, Jayhan.
All right.
I
Hello,
tried to make
everyone.
it brief because we started a little late. So
Yeah,
go ahead.
and just letting everyone know that the mission and vision of this journal club is to support the education of healthcare professionals with discussions of published research. And although we are human and filled with biases, we try to approach each paper with consistency, fairness, and transparency. So again, the purpose is really to educate, raise awareness, and engage in a professional exchange. And we're not here to recommend policy or regulatory decisions and really have no skin in the game or agenda other than just education about a very narrow, sort of focused piece of research. Just reminding everyone that this is the same link we use every month, first Tuesday of the month, for this journal club. Feel free to share it with any of your interested colleagues who would like to engage in this discussion of research papers on a regular basis. Today's featured article, as was mentioned, is entitled "The Biochemical Role of the Endocannabinoid System in the Pathophysiology of Attention Deficit Hyperactivity Disorder, a Narrative Review and Future Directions," published in the Journal of Neurochemical Research. Well, I'm going to hand it off to Dr. Phil Malloy. Why do we care about discussing this article?
Well, we visited sort of related topics before, and you can see up here some highlights from previous journal clubs. A lot of people care about the subject, obviously, if cannabis has anything to do with ADHD, either helping it or it being involved in the pathophysiology. We did look at a paper in September of 2024, that's a year and a half ago right now, on cannabis use in pregnant women. It actually showed a signal for when the kids were followed up to as long as 14 years. So the kids are now 14 years old, that exposure to perinatal cannabis increased ADHD and related disorders. And it's a subject that's of keen interest, as people who follow our journal club know. Pregnancy, use, and mental health, psychosis, ADHD, behavior issues are hot topics. That's all. So next slide. Um, I think we just said this already. There's no need to just to read it again. People who have followed the literature on pregnancy and cannabis use know that it's not trivial the number of pregnant women that use cannabis for various reasons. Okay, next slide. Here's a question just to throw out there. So why do we care? Introduce these slides. For healthcare providers who are taking care of patients or your neighbor or your relative or your kid's friends, somebody asks you about ADHD, does it have a role either in the pathogenesis or potential therapeutic role? How do you answer these questions? So we're hoping to shed light on that. And a heads up is that most of the data in this paper is preclinical and animal study, but there is some clinical information, just to let you know what's coming right now. Okay. This is background. Cannabis use is highly prevalent in individuals with ADHD and hyperactivity. Um, the current prevalence of CUD in this population, according to one recent paper, we do have references they're quoted here. It's in the notes section of these slides if anybody wants them. Is 19.2%, not a trivial number. The only randomized controlled RCT of an oral mucosal spray, everyone knows what this one is, right? In patients with ADHD, found no signal for an effect on attention, memory, or cognition. And a recent meta-analysis of 39 studies um, concluded that there were either no effect or patients got worse. And the italicized quote at the bottom of that last bullet is lifted from the conclusion of the paper. So what do we know actually? So I think most people are sort of familiar with a big high level picture that behavior, focus, integrating inputs, uh, filtering is a complex area that's not 100% worked out right now, but it's pretty clear that these neurotransmitters, one, two, and three on this slide, are involved. Dopaminergic is getting most of the attention. It seems to me, as I look at the literature right now, be interested to know what other people think about it. There are three main pathways: mesolimbic, mesocortical, and nigrostriatal, where dopaminergic is dominant in behavior, and there are signals that it is involved in the pathogenesis of ADHD, noradrenergic and serotonergic. The ECS is highly expressed in these same pathways. I would like to just say something that I know Jahan's going to say later. The fact that you look at a system like neurotransmitters from one part of the brain to the other and see that the ECS, this is a perturbation or a signature in patients who have ADHD, does not prove that what you're observing is the cause of ADHD. Could be compensatory. There are plenty of examples in medicine where there's a signature that has been suspected to be causative, and history has shown it's not causative. It's a signature. So we're not saying that the activity of the ECS, if we knew it was causing it, it would give us more validation to go down a potential therapeutic path and do clinical trials. An association is not necessarily causative right now. We do know that the ECS, as you can see from the last quote here, plays a key role in the crosstalk between these different neurotransmitter systems, is crucial in the regulation of behavioral responses. That's pretty well established. The details, the fine print, it's more unknowns than knowns. You agree, Jayhan, with what you know, right?
Yeah, absolutely. I mean, we see the ECS responding to different stressors. Doesn't necessarily mean it's causing the, pathophysiology. It might be more of a thermometer than the furnace driving the activity.
Okay, let's move ahead. Yeah, I just for fun, a couple of weeks ago, um, I use Open Evidence. It's an AI tool that's confined to medical research. So I searched cannabis and ADHD, and it's just a lot of references out there. Many of them are case reports, brief papers. It's an area that's pretty fertile right now for attention. There's more attention than there are conclusions, as everyone knows. And interpretation is confounded because we don't really understand entirely what causes ADHD. In this paper, and our view can be divided into what do we know about basic science, what do we know about what's going on in the brain, um, and does knowledge of the ECS help us even even make a postulate or a hypothesis about, I wonder if we do this, it could be therapeutic. So so what is this paper? This paper, you see what it is. It's a meta-analysis um, that ended up, some of you have read the paper, usual meta-analysis and
Hold
review.
on. It's a narrative review, not a
Right.
meta-analysis. Sorry about that.
Okay. Yeah, thank you.
So
Um,
the
It
narrative
says, it
reviews,
says narrative review. Yep.
yeah.
yeah,
And so it is those are sometimes the most fun to read because people who are in the sciences get to they can cherry pick what they want to include and examine to perhaps support a hypothesis or push in an area. I mean, Teresa, do people just do research for fun, or do they usually have like motives for doing it?
So yeah, so I always think that there's a reason behind why you're looking at such a topic, and whether it's developing, some new tool, or there's always a reason behind you doing some research, a new product that might be coming to market to treat. So yes, I don't know if everyone agrees with me, but that's just my opinion.
I think that makes a lot of sense. They could be just trying to, wherever they're based, I think these authors, you mentioned they're from Turkey, they might be trying to drum up support for more funding in this area or for approval of a product for treatment. Or again, could be a number of reasons why you might want to do a narrative review.
Exactly. Yep.
And so, but they did have a very important thing in the paper. As we know, if you attended journal clubs, probably one of the most important figures is what they included and what they excluded, whether it's research studies or human beings. You want to see how did they, what was their sort of process? Did they have a selection process? Just so you can understand what the pool of information is or the pool of their data sources. And so here we can see that they screened studies through the databases through October 2024. So that gives you an idea of how long it takes to publish research, 2-3 years sometimes. They found 20 other studies through other sources. I don't know, their best friend, the newspaper, who knows. Number of articles of accessible abstract, 60. And then they start to break it down. And then they broke down the number of articles included this review. They eventually got to 13. Any other comments on that for the moment? All right, we'll move on to the next one. So we kind of end up here when we talk about research in beginning phases. And this is where we see science struggle a lot, and that's with translation, right? So people try to take what we learned in controlled systems and apply it to the messy reality of humans. So in these animal studies, these preclinical studies, we can control just about everything. What the rat eats, who the rat talks to. In humans, we can control almost nothing as we go population scale. So we have to remember that sometimes these findings don't always carry over cleanly. And so why do preclinical studies? Because they can tell us what's possible, what mechanisms are possible. And human studies, based on animal studies, tell us what actually occurs. And you need both of them. And so I feel, again, as a basic research scientist, that this is really the heart of the paper. 11 animal studies, mostly hyperactivity models. Very little true attention-related work. And so we have to kind of be careful here, because what these studies are doing is actually fairly straightforward. They take rodents, then they pharmacologically manipulate them, manipulate the endocannabinoid system, and observe changes in activity or task performance. And that can tell us something specific, like the endocannabinoid system can modulate behavior. But it does not tell us that the ECS is causing the ADHD. This is sort of a modulation. So again, you look across these studies, you can see a few patterns. Hey, you block CB1, you block CB1 receptors, sometimes you can improve control. CB1 activation kind of produces some inconsistent dose-dependent effects. So what we're seeing here is not, is some modulatory effects, I think. And this is where the authors describe their findings as consistent, which I have a little bit of a problem with, because when we look at the rats they use, the dopamine transporter knockouts they use, and their pharmacologically induced states, they're not ADHD. They are these fragments of behavior. And so calling it consistent is a little bit generous. So what I think they're really looking at is the regulator here of the system, not necessarily the cause of it. I guess that would be my interpretation here, is that when we're looking at the endocannabinoid system being modulated, we're looking at something trying to stabilize, trying to buffer, trying to maintain balance, not driving the pathology. So I think that distinction is important as we move forward and start to look at the human science. All right, well, I'll pass it off to the MD to talk about the clinical stuff.
Yeah, sadly, there aren't a lot of other studies addressing the role of the ECS in human cases of ADHD and hyperactivity. But you'll see in the subsequent slides, Table 2 and Figure 3. There are signals, whether they are causative or associations that are not causative, whether they are potential therapeutic targets or not, is not at all clear from these two studies. So let's look at the tables in the figure right now, Jayhan. So here's Table 2. These are the two clinical studies. One was published in 2021, one was published in 2009. Small numbers of patients, as you can see. In the third column, you see the number of patients with controls. In the fourth column, you'll see what was measured. And in the far column on the right, you'll see what the main findings were. I think the main findings can be summarized by saying, compared to controls, perturbations in the ECS, either in enzymes, receptor levels, or ligands, endocannabinoids, there was a signature that was different from controls. I think that's all I can conclude from these clinical studies, these two clinical studies. It would be tempting to say, are they causative? And are these targets for potential therapeutic interventions? But postulation only right now. Do you have anything? Am I missing anything, Jayhan?
We'll talk about it a little bit on Figure 3, but I would also just mention that the pharmacology is messy here. Anandamide and 2-AG and these other conjugated endocannabinoids, they do more than one thing, right? Some people call them promiscuous. It seems oddly judgmental. I prefer maybe a multi-tool or a dynamic interaction. So if we are interacting with FAAH, we're not just affecting endocannabinoids, we're affecting the metabolism of other things as well. And if you think about FAAH activity and pain, I think I start to ponder about the effects that modulating pain can just have on behavior. So I think this brings up some interesting questions in terms of how clean is the pharmacology, how predictable is it, and what happens when there is, what happens when you throw exogenous cannabinoids into the mix? How does that interact with the system? That's my initial ponderings.
It's important because there are interventions that affect FAAH, for example, in experiment in the use right now,
Well,
looking.
yeah, like Tylenol, right? You
Yeah.
take that, it metabolizes. I believe it's pronounced acetaminophen, and
Hmm.
that metabolizes into a FAAH inhibitor. And
Yeah.
so I know we've heard some really interesting things about what Tylenol might be associated with and causing, but here I feel like it's saying the opposite, that modulating FAAH might actually provide a benefit. So I don't think the information is super consistent with when we start to look at a wider lens at these things.
Yeah, so can we move to figure 3? Because I think that
Mm.
that really
Oh,
tells the whole story. Hopefully everyone can see it
Yeah.
clearly.
yeah. So we tried to get this as cleanly as we could from the paper. So we'll take a minute with this because this is really where the paper tries to connect everything. They have a model of ADHD in the human brain, and we know it's an accurate model because there's only 2 functioning brain cells. But basically, at the core, we have the usual suspects here, right? We have dopamine circuits in the prefrontal cortex. We have systems that regulate attention, reward, behavioral control. And sort of on top of that, layered into that, we have the ECS. And what makes the ECS different from everything, everyone should know this, right, is that it's sort of a feedback mechanism, right? They travel backwards across the synapse to regulate what gets released next. So instead of driving the signal, the ECS here is adjusting it. But does this figure explain ADHD, or does it explain how the brain regulates itself more generally? Because everything in this diagram, you see dopamine modulation, you see inhibitory control, reward processing, these aren't necessarily unique to ADHD. So when you look at the top, figure A, you see like healthy, right? In theory, we can argue about what a healthy person is later, but you have balanced anandamide to AG, you have whatever is normal FAAH activity, you have effective CB1 mediated signaling, and you have this sort of stable retrograde signaling, which I think plays into this idea of balanced endocannabinoid activity. And in ADHD, they are hypothesizing that there's decrease in anandamide or 2-AG, there's an increase in FAAH activity, decrease in CB1 signaling, changes in glutamate and GABA, and again, disrupted retrograde signaling. And when we start to look at these pathways, we're looking at sort of how these systems are modulated in different disease populations. And then they talk about disease-based therapies, some of which they haven't really talked about yet in the paper, like the TRPV1 modulators, most notably activated by chili peppers. I don't recommend doing a jalapeno-based clinical study to treat symptoms of ADHD. But what's interesting here is CB1 antagonists. And so I think that I'm starting to see inconsistencies here. Why would you want to block CB1 and inhibit FAAH? So you're increasing the availability of a lot of stuff that stimulates CB1 receptors while blocking it. Interesting idea. Maybe it's pushing their pharmacology to other targets is the idea. Couldn't really say. We do know that cannabinoids and endocannabinoids will act on the TRPV1 channel, so that could be something they're trying to push there. So
So
this
yeah,
is,
so I think that we try to go through the next few because we might come back to this in
yeah.
discussion. I'd like to hear what the audience has to say about some of
So
this.
we can come back to this. I
Uh,
would say this is a
I
map
think Dr.
of
Kamen was
influence.
just
Oh,
gonna,
yeah.
Dr. Kamen was just gonna, hi.
Hi guys. I just signed on. I think my observations are, that on the healthy side versus the ADHD side, it's glutamate and I'll decrease in GABA, which is the inhibitory uh, paraprotein that's circulating. And this lack of, the inhibition is what characterizes ADHD. They're just all over the place. So it's that out of balance, glutamate being a very prominent component of nociception or pain, being more evident, and the lack of inhibition in that ADHD, it explains the behavior. What happens when you throw exogenous and cannabinoids in there is another story, and that we don't know. Yeah.
Thank you. That was great information about the glutamate and GABA signaling. Um, and again, I think of this as a map of influence more than like a map of disease or pathology. So we'll move on to some of the primary conclusions of the paper. We can argue about some of these points or discuss them. One of the points, primary conclusions, is that the animal and human models are consistent. I have issue with this, but we can discuss some of their other points. Again, there's growing evidence to suggest that the ECA plays a central role in neurodevelopmental processes and dopaminergic modulation, cognitive control, reward processing, and inhibitory control, stress responsivity, and neuroimmune regulation. All these domains that align with the neurobiology of ADHD. I think most of us will agree that yes, there are these fragments of ADHD that the ECS responds to 100%. I am on board with that. And, there's these biochemical patterns that folks are seeing linking the ECS to these ADHD-related behaviors. Again, suggesting potential mechanistic relevance. Gosh, you manipulate a rat with a pharmacological assay. Gosh, that looks a lot like a symptom of ADHD. Human and animal models show ECS dysregulation represents a biologically plausible and potentially influential component, warranting more systematic and developmentally informed investigation. Agree definitely with that last part. I even think there needs to be more research just in what is ECS dysregulation. Your ECS should change in response to different challenges and stressors. It shouldn't remain constant all the time, right? The point is to respond and help bring you back to an appropriate level. So what does ECS dysregulation look like? What are those ranges? What are those buckets? Indirect enhancement of endocannabinoid tone, FAAH, MAGL lipase, reduces hyperactivity and improves inhibitory control. Great stuff there. The balance of CB1 agonism and antagonism and ECS dopamine interactions appear to play a role. I think this is sort of the yin and yang relationship with the endocannabinoid system that we need to always remember, is that sometimes there can be overactivity of the system, there can be underactivity, understimulation, overstimulation, and balancing that will be important. ECS signaling may also influence the profile of established ADHD treatments. I don't want to say drug-drug interaction there, but it does sound like they're suggesting that. But anyone is welcome to start commenting on some of these conclusions. We've got a couple slides left, but if you have any comments or questions, put them in the chat. Or if you'd like to go back to any of the slides, we're happy to do that.
Yeah, can we just go to the summary then if you're at the end of that? Like just
Yeah, we have just a few. Yeah. Again, many of their conclusions kind of overlap with, hey, there's insufficient evidence to determine whether exogenous cannabinoids improve or worsen ADHD symptoms. They discuss more clinical things. But let's go to, in their own words, the authors summarize their models. Teresa, would you like to discuss?
Sure. So so both of these points I think are really pretty important, and they resonated with me. And that's the heterogeneity across their ADHD models in their animal studies. They each captured something different, a different symptom or domain, which complicates the interpretation. And so that's similar to for those of us who do, clinical trials, you're giving someone, an SF-36, which is one type of patient-reported outcome, and you're using some other specific disease like an ADHD, quality of life type thing, and then you try to say that they're similar, but when in fact they're not, when they look at the little domains in between. So this I think it makes it challenging to come up with any kind of overall interpretation. And then the other piece is the translational piece. So there's methodologic and conceptual constraints in the preclinical findings that can actually be translated to the human studies. So I think that's really what I found to be interesting in this paper. And I would like to know, I know it's gotten a lot of press apparently. So it'd be interesting to hear from the audience, like what they're thinking about this paper.
And don't all unmute yourselves, at the same time. But you're welcome to, Mark Viner, Dr. Viner, please. We welcome any comments or questions.
Oh, can you hear me okay?
Yes, sir.
Yes.
Okay, it's okay study, but it's leaving out new things that are important, like the dopamine's okay as far as locomotion towards or away things impulsively, but didn't they not comment on the noradrenergic pathways? Because CBG, I think we got to look at alpha 2 agonists because they're used now much more popularly in medicine. There's medicines in psych, I'm a psychiatrist, board certified, for agitation associated with bipolar and schizophrenia. It's called IGAMLI. It's an alpha 2 agonist. And all of the groups like meds for ADHD, like guanfacine XR and clonidine XR, the ones that try to get the hyperactive impulsive presentation, are alpha 2 agonists. And CBG is a classic alpha 2 agonist. So I'm hoping to see the results of the Kentucky doctor who's doing a clinical trial, clinicaltrials.gov. The results should be out by the end of the year on CBG for ADHD, hyperactive impulsivity. It's an alpha 2 agonist. And of course, we got to know about alpha 2 agonists because this illicit fentanyl, the Drug Enforcement Agency just put out a warning that they're cutting fentanyl with alpha 2 agonists like clonidine that's used, for aid that causes bradycardia, hypotension. So it makes the overdose with fentanyl even more lethal, and the withdrawals, tachycardia and hypertension, even worse. So, the dopamine models, they left out the other pathways and the nodal connectivity of the brain when you talk about motor, impulsivity, hyperactivity. So, these are just a few things I noted that it really wasn't, they didn't really address any of those. Okay, thanks.
Yeah, those are great points, Mark. And it reminds me of the study about CBG that Ethan Russo was a part of, and they found improvements in memory and other aspects that weren't necessarily related to anxiety per se. But I think that the CBG as a factor in why maybe people are observing at times cannabis works and cannabis doesn't. Maybe it's because of this pharmacology in these products where sometimes there are significant amounts of alpha-2 agonists that are creating a beneficial response. It'd be a very interesting study to control. And yeah, I really like that hypothesis about the cannabigerol CBG and looking more closely at it for specific effects. They were looking at it for anxiety for a while, but maybe its true therapeutic utility might lie elsewhere.
Yeah, thanks, Mark, for that. And keep us posted on that. May be a good paper to bring to Journal Club, right?
Yeah,
As a
absolutely.
follow-up. Yeah, yeah. Maybe a year from now, if it's reading out in about, you said it's reading out soon, maybe it'll get published soon, a year later.
Well, we have a little bit of time left. Are there any questions about the article? Would anyone like to go back and look at another figure? Do they have their own observations, concerns? Are there about cannabis and ADHD or cannabinoids and ADHD? Maybe you have questions about just some of the scientific principles used in narrative reviews and animal research or clinical trials? Happy to discuss those as well.
Yeah, let me throw something out. Do you think ADHD is just too heterogeneous and there's too many phenotypes out there to do a clinical trial in? I mean, are people just too different?
I think there is an aspect of that similar to pain and what people want to see resolved. If you think about the different types of pain, there's acute pain, there's chronic pain, and maybe Len, has a lot of direct experience with this, could comment on maybe something analogous where there's all these subcategories, and I think terms like ADHD seem to get thrown around a lot. Like I don't think I can go more than 5 minutes doom scrolling without seeing someone saying they have ADHD because they like to organize all their puzzles or something like that. But
Not to be disparaging in any way to my psychiatric colleagues, these are diagnoses that are determined by consensus panels. There's no biomarker other than what we're seeing today here in pain. Similarly, that biomarkers are absent. What we do know is, again, that's a little more about neurophysiology and the drivers of behavior through these observations, and that's more of a objective evidence track for pain. What I'm actually curious about now is how the endocannabinoids work on G protein coupled transport, and the pain medications all work on G protein coupled transport, and I think we're at the basic, we're at a better understanding of how critical this is in terms of what functionally the outcomes are, and when we're adding the effects of exogenous or phytocannabinoids and interfering in that G protein transport, we're probably seeing a lot of elements of receptor activities that are affecting behaviors. So I think to Phil's point, there are many phenotypes, and in pain, I think it's always up to the individual's perception, their early experiences with pain, and what it generates in terms of fear and threat to the organism, and how we respond to fear and threat and turn that into chronic pain, because we can't ever get over the concept is probably based on this neurobiology of these transport systems that move the proteins that are endogenous to our bodies as well as the exogenous ones, and we're still playing with that. That's as much as I can offer right now.
That's a great point, because there are issues with intracellular trafficking too, with cannabinoids affecting not just the outside of the cell, but the inside as well, and some of those dynamics. And I think you brought up a really great point that also makes research difficult to translate from animal studies, is that humans perceive things a bit differently in their environment, and that plays a big role in some of these conditions, is the environment and the complexity of that environment. And I really like that comment. Oh, Dr. Liz Thompson, PhD. Welcome. You're live on the Journal Club. All right.
So Jayhan, Sanjita shared a paper, and I actually am familiar with the UK Medical Cannabis Registry. I did not know that they just published something on, or they did publish something on ADHD. It might be worth having a look at that. So thanks for sharing.
Yeah. Very cool. I wonder how much of that information made it into this study that we're talking about.
One thing I find very important with the cannabinoids and ADHD is the thinning of the brain. You'd see the cortical thinning with ADHD in contrast to autism, where it's too thick. And then the CB1 receptors in the cortex, mainly the frontal, where there's a lot of CB1 receptors, relatively speaking. And early in brain development, it's involved with pruning. The cannabinoids are involved with normal pruning. So I always contrast autism with the ADHD, because you get this great cortical thinning. The brain doesn't develop completely with the ADHD patients, I find. So, yeah. Oh, and then finally, to comment on someone's question, there's some articles, some doctors are thinking ADHD is like a trait. It's not a disorder. So you were saying there's a variety of presentations like depression. So anyway, some people think it's like a trait with various degrees in everyone. It's
And
fascinating.
that makes more
Yeah.
sense, right? I mean, it's almost seems to be that would make more clinical sense when you're trying to
Yeah.
diagnose someone. Any other comments? Oh, did Dr.
I,
Thompson? Oh, here she is now.
yeah,
She's back again. Okay, go ahead. Oh,
Oh,
there you go.
sorry, my microphone was muted. And I had to go and fix the settings. Anyways, I think something really interesting that doesn't come up a lot is, can you hear me okay?
mm-hmm.
Yeah, is we talk a lot about the receptors and the transporters and the ligands and all of that, but I think one thing that was missed here and gets often missed is the role of bioenergetics in the brain. And I think that the endocannabinoid system plays a big role. We're running clinical trials studying concussion and how we can use cannabinoids to possibly reduce the neuroinflammation in the brain from contact sport concussions. And we know that concussion really is driven by the secondary cascade of the metabolic influences after the primary injury, so the biochemical. And we know that cannabinoids can influence that. But it seems like even in ADHD, if you look at the bioenergetics of the brain and how we can influence that, a lot of the symptoms that we see in concussion or ADHD or any of the pathologies that are grouped into these groups, if you look at how the bioenergetics and the ability to get the proper amount of energy to the brain to have it properly operate, I think we missed that. It was missed in this paper and it gets missed often. But I think because we have CB1 receptors on the mitochondria of the cell, which is the organelle that creates ATP, we know that we can hit those CB1 receptors on the mitochondria, which increases the ability to produce ATP correctly. And that can help to shift the bioenergetics of the whole system back into a place where you're not going to see the outcome. But in concussion, we're going with that theory. And so in ADHD and other potential things that come out as maybe mental things that you see in people as behaviors can be reversed. And we know in bipolar with mania and stuff like that, a lot of times when you look at the glutamate and things like that, the excitatory, of course, in concussion, it's an oversecretion of the excitatory neurotransmitter glutamate and the inability to put the brakes on your brain. And so when we use cannabinoids to do that in concussion, we're able to sort of re-regulate that. And on concussion, a lot of the times, to your point, Jehan, where you see everybody putting everything in the category of ADHD, concussion is very multimodal. Like it doesn't look the same. You can have 10 athletes that have been diagnosed with concussion, and that's a separate topic of how and what are we using to diagnose concussion. But, it's thrown into this huge category and the output looks different for all 10 of those people, but all 10 of them would say they have a concussion. And so I think when you look at ADHD, it could potentially be the same thing. It's, you have multiple things driving multiple outputs. And so they're being put in one category, usually for CPT coding, so that we can code it as one thing for insurance. But, I think as researchers, it's important to look at what are you actually looking at, within the context of the definition of what clinicians are putting it into, categories because they have to. But when you're really looking at mechanisms, I think bioenergetics gets missed a lot. And I think the endocannabinoid system plays a huge role in bioenergetics in the brain, which can come out as, 100 different things because it affects your autonomic nervous system. It affects, mental personality. It affects all the things we see in concussion. If you think of anyone you've known as having a concussion, they have all kinds of different symptoms. So that's just one thought that I had as it pertains to ADHD. I look at things that come up in the mental sphere just because we see someone with a concussion would potentially put themselves, say, I have ADHD temporarily, because they feel that way, but it's being driven by the concussion problems, and then you reset it and it goes away. So I don't know, but that's just my thoughts on that.
Thank you so much. That was fascinating.
Yep.
And so I really like it because I like these themes that are developing across different conditions. So, it's not my imagination when we see these things being very different for the individual, whether it's pain, ADHD, concussions. So it's kind of comforting in some way to know that this chaos is normal when it comes to diagnosing or assessing these things. And that's part of the challenge with science, is pulling on these different threads. And Dr. Kamen, I also liked your comment that we don't have, a blood test for many mental health conditions. There isn't like a single gene that predicts diseases related to ADHD that I know of, anyway. And I thought that was a great point as well, and the challenge of studying and assessing the quality of research in these areas.
Yeah, no, thank you. I think that these nuances are very important to take into consideration when you are doing research. Any other comments? Dr. Kamen? Finished? Everyone? Oh, so should we just go to the next month's journal club?
Oh, yeah. Real quick, strengths and weaknesses. Again, we'll say paper. I'm in agreement with Dr. Malloy on this slide. Clear questions for focus, solid methodology. There was a review article with no new data of studies. I felt it downplayed some of the inconsistencies and competing explanations. And pharmacology in this area is messy, and I feel that was under discussed. The next journal club will be May 5th, and we'll be celebrating a different way. We'll be celebrating with high-dose THC and a discussion of psychosis, a recent article. So if you want a if you want to take a break from your margaritas and join us in the afternoon for a fun-filled discussion of a new paper that has come out.
Or
These are
bring your
always fun to
tacos, or
discuss.
bring your tacos for lunch,
Yes.
or your nachos or something.
Taco Journal Club Tuesday will
Right,
be.
tacos, nachos, something, right.
Marisa, we didn't put the reference on here. Do you remember the author or journal? Just
Yes, I have the paper open. Hold, please.
in case anyone wants to look at it ahead of time, we neglected on this slide to put the
Yes,
reference
so
in case anybody.
it's the Annals of Internal Medicine. I can just pop it in the chat, I think.
All right. Okay.
And and we
I
will post the announcement on LinkedIn as well as send it to the other medical societies that share these news, the news of our journal clubs.
Yeah, I'll see if I can do this.
All right.
Everyone can't hang up yet, though, then. All
Well, for
right.
those of you who are here, we want to say we appreciate all of you for your time and attention to helping create
Hi.
this lovely session today.
Thank you, guys. This is